All right. Hello, everyone. Can I just get everyone's attention, please? All right. Thank you so much for joining everyone today. This is our last educational event for the year, which is crazy. I feel like the year has just flew by. So today we have a research seminar talk for you folks today on how physical activity can enhance our well-being across many domains of health. My name is Lily, and I'm one of the program assistants for the BC Brain Wellness Program, and I will be your MC for today. Before we dive in, I do want to take a moment to acknowledge that the BC Brain Wellness Program here at the Javad Mofe Center for Brain Health resides on the traditional, ancestral, and unceded territories of the Musqueam peoples. As a settler here, I think it's really crucial to understand the connections we have with the land and to continue to honor and nurture these connections. It's truly a privilege to be able to offer our program on Musqueam land. And I know we're probably joined from various locations today, especially for those on Zoom. So we have a resource that we will paste in the chat that will allow you to learn more about the history of the land that you may be on. And we have a QR code for those in person. All right, so we are super grateful today to be having Dr. Eli Puterman. So a little bit about Dr. Puterman. So Dr. Puterman is an associate professor here at UBC in the School of Kinesiology. He has been the Canada Research Chair in Physical Activity and Health since 2015 and was recently elected as a member to the Royal Society of Canada College of Scholars, Artists, and Scientists. Dr. Puterman's research program focuses on how non-biological processes, including psychosocial and behavioral factors that impact mental, physical, and cellular health, with the goal of revealing new insights into understanding treatments of disease and mortality. So for today's talk, Dr. Puterman will present information about the wide scope of stressful experiences that impact our health and well-being, the pathways through which these occur, and what we can do about it. And specifically, he will present some study findings showing how movement-based activities can promote health and well-being, even deep within ourselves, as in adults undergoing adversity throughout the lifespan. And I would also like to take a moment to thank all of our program donors for supporting the BC Brain Wellness Program. It is because of your generosity that we're able to continue to really support these classes and educational events. So as an entirely donor-funded program, thank you so much for your ongoing support. And a couple of housekeeping notes before we begin. If you have any questions for Dr. Puterman today, feel free to type them in the chat for those on Zoom. And if you want to ask your questions privately, you can just send it to us privately on Zoom as well. And for those in person, if you have a question, feel free to just raise your hand at the end. And when you do, there's a red button at the front. So if you can just press that to unmute your mic and ask and then turn it off when you're done, that would be appreciated. And we will have a Q&A session at the end. So yeah, and reminder for those on Zoom, if you can please keep your mics muted throughout the talk. And if the Q&A session runs over, if you're online, feel free to just stay on or log off as you see fit. And for those in person, likewise, feel free to leave if you need to. And the session is being recorded and it'll be made live on our YouTube channel in about 2 weeks' time. And lastly, if you're looking for something festive to do, we have a in-person music workshop here at the Center for Brain Health on December 12th with Cynthia. And next year we have for January Wellness Wednesday, we have Dr. Jennifer Lutz will be coming in from UBCO, who will be talking about setting goals and health behavioral changes in the context of New Year's resolutions. So registration for these are live and the links will be posted in the chat. And without further ado, I will pass the mic to Dr. Puterman. Welcome.
Hi, everyone. Can you all hear me? Yep, awesome. I will just get this all organized, get my coffee in place, my water in place. Okay, I see, oh yeah, thank you. I see a number of people are in the room, just a handful, but we do have a significant number of people online. So if I'm assuming everyone can hear me and that they will let you know if they can't. Today I'm giving a talk on life stress, caregiving stress, and health, the importance of self-care with exercise. Maybe I just won't look at this, sorry. This is your notes. So I'm like, this is confusing me. Okay, perfect. So I am a health psychologist and by training, clinical psychologist master's, health psychology PhD, and I intersect between the areas of stress psychology, stress physiology, and exercise behaviors in order to support people living with chronic stress in their lives. I've completed several different kind of intervention studies during the pandemic with healthcare workers and regular Canadians who were not using their bodies as much, but to try to help them manage their stress levels and their depression levels through exercise. I'm not touching on that at all. I'll be focusing primarily on stress and its impact on health and well-being, kind of unpack what stress means, unpack what stress means for the audience, all the different ways that we in psychology think about the concept of stress, how stress impacts health, what are the pathways, and then highlight some research studies that we've done in the past with family caregivers showing radical changes in physiological markers of health, cardiovascular health, and also psychological states and traits. So with that in mind, I will start the lecture. First, I do want to thank and acknowledge the serious contributions to my career and to my thinking by many of my mentors from when I was at UBC in my grad program with Anita DeLonges in the middle, professor in psychology, and also the team at UCSF and elsewhere at Columbia University, Alyssa Eppel, who was my primary supervisor during my postdoc years ago, who a lot of the work I'm going to present, I've done a lot of collaborations with her over the years, Elizabeth Blackburn, who's been a Nobel laureate, Nancy Adler, was at UCSF until she recently passed, and Richard Sloan and Kirsten Johansson. This is my team. I love them. They're a fantastic group of students, grad students. So I kind of talked about what my purpose of my talk is. So let me just get into right away the lifespan versus health span. I'm wondering if people in this room can give me a definition of what you consider to be lifespan and what you consider to be health span. And if you do want to speak up, please put on your microphone, or if there's anyone in the audience on Zoom. Anyone want to tell me what you know of the word lifespan? Anyone? Do I have to call on someone with a white hat on who's in the middle of drinking water? Yeah.
Yes.
Okay, yes, yeah. Thank you. You saved someone in the audience here, so perfect. Oh, was there someone who's coming on? Okay, well, I'll start the definitions. How's that? Lifespan is the extent to which people live, how long people live for. But the health span is how long do people live healthily for before there's decline in health. Those are very two strong different concepts. There's been a lot of research on how long people live and this emerging idea that we need to not just be focusing on how long are people living, but how long are people living healthily and well. When you think about it over here, you can see that the health span from poor health span to optimal health span in general, as people are aging in childhood, there's really no differentiation. Most people are living healthily, but as we age, as people progress through middle age and advanced age, that there are a group of people who there is the beginning of some poor health or some ill health, the development of diseases, development of disability, and that can lead to, or that typically ends with an earlier lifespan than individuals who mostly live robustly, healthily, with less illness, less disease that kind of impacts their capacity to live on a daily basis in a healthy way. and in a well way. And that really this idea is that there is a goal out there within medicine, and especially preventive medicine, is to extend the number of years in healthy life so that people are able to enjoy their social connections as much as they typically used to when they were younger, in order to be able to move their bodies, continue their activities of daily life living for as long as possible. When we think about the health span and lifespan, there was a great, every year the Lancet, which is this large scientific journal, they publish, how long are people living globally and how long are people living healthily globally. And they've been doing this for, I think, 30 years at this point. And every year that they publish it, they'll have a different focus. So they'll have a focus on by country, by region, by sex or gender, by income levels. And they'll, every year there's like a new focus of what they're trying to unpack. And in 2013, they published this article that they do every single year. And they were reporting on the life expectancy that people have if you were born in 1990 versus born in 2013. What is the likely life expectancy? And also what is the likely expectancy to live a healthy life, not just length and additional, not just like how long people are living for, but how long people are living for healthily. So I'm going to unpack one of these because it's a bit of a complicated figure, but I'm going to unpack it. So let's look at people who in this figure that people who were from Japan, and the life expectancy and the healthy life expectancy for individuals living in Japan from 2013. So if you look here, in 2013, the left side is men, the right side is women. So you can see these over here. In 2013, the end of the lifespan for individuals who were born in Japan, born in 1990, had a life expectancy, men had a life expectancy of 76 years old, and a life expectancy for women was 82 years old. The end of the health span, however, was a bit earlier. So how many years were they living healthily before they had years living with disease and disability? That's 68 for men and 73 for women, suggesting that 8 years of a man's life in Japan born in 1990 are years living with ill health and disease. And similar for women, there's a 9-year discrepancy. Fast forward to people born in 2013, an expected lifespan was 80 and 86, so 80 for men and 86 for women, increasing the lifespan for men by 4 years and for women by 4 years as well. You can applaud that. We can say that that's great. We've done some really great progress in medicine. But if you look at the health span, you now see that for men, the health span, or the number of years with disability, let's say, has gone from 8 to 9 of their last years, and for women has gone from 9 to 11 years of ill health. So that we're even though we've increased lifespan dramatically over the past century, it doesn't necessarily translate to also increasing how long people are living healthily, and people are living 9 years with disability, disease, and potentially some negative impacts on their daily lives as a result. I'm going to skip the United States data because we're all mad at them right now. Let's, when we think about what are the factors that people discuss that really impact the health span and the lifespan, there's a significant amount of research that really has highlighted these structural determinants and personal determinants of health that really impact longevity and especially healthy years. When we think about, this is a slide from the World Health Organization's health equity framework that really tries to break down what are the social determinants of health and address what they all are, and trying to develop strategies within the governments across the world and within the World Health Organization on how to address them. But if you look at some of the factors that impact, there are socioeconomic and political contexts, so the governance, macroeconomic policies, social policies such as housing, labor markets and lands and so on. There's individual contexts like an individual's employment status. So there's a lot of work showing that people who are blue collar versus white collar have a lower health span and lower lifespan. People's occupation, their income level impacts health span and lifespan, and also is part of this health inequity. Gender, ethnicity, and so on, those are all factors that play into health equity and, well, current health inequities that we need to address in order to reach health equity. Their mindsets and behaviors, which is kind of what some of the areas that I focus on, is so what are the material circumstances? What are the quality of the housing that people have? What are their work environments? Psychological factors like early adversity, being exposed to early adversity has a significant impact on our health and well-being. Others, so social support, how socially connected you are to other people has a strong impact. The behaviors that we engage in, our physical activity levels, our smoking status, our alcohol use, and many other factors also play into health, our health span, our lifespan, which are also determined by some of the socioeconomic and political contexts, as well as by individual contexts of work status and so on. And then there's also health systems that also impact. So the distribution of health support and medicine across different countries and within different countries and across classes and so on. So within that, I'm going to focus mostly on mindsets and behaviors, psychology, stress and behavior, stress and physical activity in this talk. But within the knowledge that most of that is typically shaped by many of these social determinants of health. So I'm going to go back to the audience and see if anyone has a definition for stress. Yes. Thank you. I think it's on. Yeah, now it's on.
Okay. So some of the classic definitions are disturbance of homeostasis. I'll go with that.
Fantastic. That's a great Cellier model. Hans Cellier in 1936 was the first to use the word stress in a scientific paper and declared it to be the disturbance of homeostasis, where some sort of event occurs physical, in the beginning it was physical, so physical pain, cold, extreme heat, and so on, would perturb your body, and that was physical stress, with physical stressors impacting physiological stress markers, and that it's a mostly adaptive process. When I am giving a talk, I'm going to have these changes physiologically and in my brain in order to be able to manage this talk and be able to engage in it with this room in an adequate way. So those are adaptive processes, fight or flight. If someone here came to attack me, I would have to respond in some way and activate my physiological system. So a stressor is occurring, and then I would have to respond. That's very important, fight or flight. But there's a lot of research showing that over long periods of time, when people are exposed to too much stress, especially psychological stress, they end up having perturbations or changes that are more long-lasting. So I'm going to show you a definition of stress, or a model of stress, that we proposed when I was at the University of California, San Francisco. It's pretty hefty. Stress is a multi-dimensional construct. So if I think about an acute stressor that I'm currently engaged in, so that's this acute stressor within here, we have these cognitive appraisals. So are we seeing this event as something benign? Is it a challenge? Is it stressful? Is it threatening or not? And if it's challenging or threatening, I have different physiological responses. We activate the sympathetic nervous system for fight or flight. If it's threatening, we also, in addition to the sympathetic nervous system, we activate the hypothalamus, pituitary adrenal axis. So we have this release of cortisol because we need some additional glucose, because that's the primary role of cortisol, is to help release glucose stores so that we can continue within this threatening environment. And we also have these emotional responses to challenge and threat of like fear or calm and so on. When you think about how this happens on a daily basis, there are things that predict it. So how people process information as events are occurring play into whether we appraise things as threat or challenge, so our mental filters, which are then also shaped by our context, our predispositions, our genetics, how our family raised us, some of the experiences that we had in early childhood, how much social support we had. Those shape our mental filters. And as a consequence of the activation of the physiological stress systems, if it's repeated too often, we have what is conceived as or conceptualized as concept of allostatic load. As you activate these systems too regularly, too often, and they can't return back to homeostasis or come back to your maintained kind of state, they are now at elevated states or sub-level states physiologically that remain more permanent. So instead of having always coming back to your regular heart rate following a stressor, some people have this constant activation of their heart. So there's increase in their regular baseline heart rate, and that's driven by physiological mediators. And the more allostatic load people have, the more likely they are to have biological aging and have early disease. There's a really great amount of research out there really highlighting that perceived stress is associated with incident coronary heart disease. So these are studies. This is an older meta-analysis from 2012, but published in the American Journal of Cardiology, really highlighting that people with high levels of perceived stress have an increased likelihood, and across all these studies, it's 7 or 8 years in the future, have an increased likelihood of developing incident coronary heart disease by 27%. So a 27% increased risk over time. When you look at mortality, and I'll just focus on this one, it's very small script, it seems, is that perceived stress and mortality. Now, of course, this is also perspective because you don't have mortality data on someone who can also report perceived stress. But if you look outwards, and this is data for, I think, 8 years away as well, is that there's an increased risk of 30-something percent, 25% in mortality 8 years later for those individuals who have high stress. When we think about how stress affects your body, there's respiratory and cardiovascular problems that occur, reproductive problems that occur, musculoskeletal system issues, central nervous system. There's research really showing that stress can impact depression, impact the development of mental health problems, but also impact the pathogenesis or the development and the progress of Alzheimer's disease and other types of neurological problems or disorders. There's also cardiovascular system problems, as I mentioned, and then digestive and endocrine system problems that arise from people who are undergoing too much chronic stress for too long. Some of the research that we've done in the lab has focused heavily on the experiences of early life stress and how they drive some markers of health and also early mortality. I'm going to show you one on a telomere length. This telomere length is this marker of cell aging. So at the end of our chromosomes, we have these repeated DNA sequences of TTAGG, and they're repeated hundreds, thousands of times within everyone's replicating cells. And the purpose of them, they used to think that these were like, they were junk material that was just at the end of chromosomes, that really coded for nothing, useless. But what they realized, actually, before I get into that, as you are familiar, potentially, is that our DNA is used to create proteins. So it goes through transcription and translation to code for proteins. And what they discovered is that the DNA that's in your chromosome that's coding for protein is quite special. It's quite unique, and you don't want to cause any damage to it. So what the telomere's purpose, they discovered, is literally there to be the junk material so that every time a cell divides and doesn't always divide with being able to replicate the ends as long and keep the ends of the chromosomes long, there's this problem every time a cell divides called the 3' end problem. And every time the cell divides, it loses a bit of the DNA that's at the end of the chromosome. So if you're going to replicate and lose some DNA, you better not lose DNA that's really important to producing proteins, right? You want to lose DNA that's not that important. And that's what the telomere's purpose, part of the telomere's purpose, is to really ensure that during cell division and also during exposure to radiation, that it encapsulates and also protects the rest of the DNA that's really important. But over time, it does get shorter to a point over here where it reaches a critical point and the cell enters cell death. And what they've shown is that there is a lot of research up until before this research showing that early adversity and other types of adverse experiences, caregiving, being a caregiver, having high depression, having unemployment, long-term periods of high stress in someone's life is related to short telomeres. This is a study that was probably one of the first using national data. This is in 5,000 people. We tracked, it's a study called the Health and Retirement Study. They had all these exposures that people had to early life stress, and we wanted to see whether it's related to telomere length in adults who are on average 70 years old. So this is really about this long arm of early adversity being able to impact us later in life. This is the HRS study. This has been ongoing since 1992. And over that time, they collected all these questions on childhood adversity. So did you at some point in your early life have to relocate due to financial difficulties? Did your family receive financial help? Was your father ever unemployed during your early childhood? Did you yourself have trouble with the police before age 18? Did you have to repeat school? Were you physically abused? And did your parents use drugs or alcohol excessively? They also asked questions over time about adulthood exposure. So experiencing the death of a child, experiencing the death of a spouse, experiencing a natural disaster, firing a weapon in combat, ever had a partner addicted to drugs or alcohol, being a victim of physical attacks, ever having a spouse or child with serious illness, being on Medicaid, so some financial questions too, being on Medicaid, food stamps, or being laid off, so unemployment exposure as well. And what we wanted to see was whether any of these individually predict early shorter telomeres later in life. But not only that, but what happens if you accumulate these exposures? So it's not about one exposure versus another, but accumulation of more and more of these types of events in your life, predicting an increased likelihood. And what we found was that primarily across all these different types of exposures, none really, except for trouble with police before age 18, none of them really independently predicted the likelihood of having short telomeres versus being categorized as someone with longer telomeres in this cohort. When we added them all together, and you'll see here this word that just popped up, total lifespan adversity. So you added up all of the ones from childhood and adulthood, and what we found after you adjusted for BMI and age and everything you can think of under the sun, when we just for literally everything, that there was this increased risk that for every one additional event that could have occurred or adverse experience that could have occurred to these individuals across their lives, there was an increased risk of having short telomeres by 7%. And when we teased apart the childhood versus adulthood, it seemed that childhood adversity seems to be driving, and that's where this comes in. This is the adulthood one. This is the childhood one. They're not crossing this one. So that means if it's not crossing the one, which is here with childhood, it's significant. So childhood exposures seem to be deriving this idea or the relationship between early adversity and shorter telomeres later in life in a sample of participants average age is 70. And when we teased it apart even further, it was really those psychosocial experiences of getting into trouble with police, having parents who were physically abusive, parents who used drugs and alcohol, and having to repeat school were the ones that were driving, not the economic factors, but the psychosocial factors. So that switches me to, okay, now that I've talked about how depressing life can be when we're exposed to chronic stressors, let's talk about some risk factors that we can potentially help manage. So we know that, let's say, this is from, what's the organization called? The heart people here in Canada. Anyway, the name's completely escaping my mind. Sorry about that. But if we think about these lifestyle risk factors, that stress and all these other ones, unhealthy diet, physical inactivity, unhealthy weight, excessive alcohol use, smoking, they're all impacting our health, but stress also impacts our engagement in many of these different types of behaviors. So we can't really see physical inactivity outside sometimes of how when we're stressed out, we are less likely to be physically active. When we are stressed out, we're more likely to be driven towards unhealthy diets because those types of sugary and salty foods, they stimulate areas of our brain that calm down our stress responses. So a lot of this is natural. Or naturally driven or unconsciously driven, many of these unhealthy behaviors, especially when we're under stressful conditions. But I'm going to flip it in this work where really we can also use physical activity to actually bolster our adaptive stress responses and dampen our negative stress responses. So here is a cartoon that says, I'm prescribing exercise. Think of it as a stress pill that takes 30 minutes to swallow. Hippocrates said, walking is man's best medicine. Many of you probably know it from literature or have heard that there are many health benefits of physical activity. It reduces dementia risk by 30%, hip fractures by 68%. So people who are physically active are less likely to have hip fractures. It reduces depression by 30%, all-cause mortality by 30%, cardiovascular disease by 35%, type 2 diabetes up to 40%, and colon cancer and breast cancer by 30%. So there's how much activity is the amount of activity? I would say any activity is better than no activity. 1 step is better than 0 steps. 100 steps is better than 90 steps, right? But when we think about what the governments recommend, they've created in Canada this 24-hour movement guideline for older adults as well as midlife adults and children. And what they all state, well, children is different from adults, but the adults, whether you're young adults, midlife adults, or older adults, typically recommend up to, minimally 150 minutes of moderate to vigorous physical activity, and also some muscle strength training, so resistance training, some sleep, that they recommend 7 to 8 hours of sleep, and also sedentary behavior to do less than, limiting it up to 8 hours or less per day of keeping our bodies sedentary. Many of us are sitting at work, so in addition to those work hours, maybe an hour more of sitting. So those are the recommendations for physical activity, sleep, and sedentary behavior. I am very heavily interested in, can physical activity support people who are chronically stressed in moving their bodies, in mitigating some of the negative consequences of stress. And I'm going to highlight one study that I did several years ago. I think it was done 8 years ago, and then we published a series of papers as a result of this study. And I'll focus on these 3 studies, or these 3 manuscripts that come out of this one study. So it was the Fitness, Aging, and Stress study. It was about improving movement behaviors in family caregivers. So family caregivers of Alzheimer's patients or individuals with other types of dementia are, compared to other caregivers who are supporting other individuals with other types of diseases or chronic health conditions, family caregivers of Alzheimer's patients have an increased risk of depression by, I think, twice as much of a risk of depression compared to other caregivers and other individuals who are not caregiving for someone with Alzheimer's disease. They also have an increased risk of cardiovascular disease by 60%. And they also have an increased risk of earlier mortality. And a lot of that has to do with the what they call is caregiver burden, the difficulties on a daily basis, the lack of sleep that occurs, the stress that's coming from it, and also the kind of that social bond that you have with either your partner or parent that is quite strong, and then you're losing that person because their personality is changing somewhat, and you're losing that social connection as well. So I proposed at some point over a decade ago to the NIH to complete a study when I was in San Francisco, to complete a study where we are providing exercise support to family caregivers to see if we can reverse or change some of the negative consequences of caregiving. And I was told that the lives of caregivers, not by the NIH, they supported it, but I was told by a big caregiver researcher that I will never be able to get caregivers to move their bodies more. It's impossible. They're so burdened. It's impossible to even, he's like, I gave them bikes once in their house and it's impossible to do this. I'm like, I'll make this happen. You've just challenged me, and I take up challenges to make sure things can happen. So what did we do? We I went to a lot of Alzheimer's groups in the Bay Area when I was there. I went to 4 or 5 different Alzheimer's groups, talked to them about the study, but also asked them, like, what would you need additionally to what I'm describing? What would you need additionally to move your bodies more? And they had mentioned, if you having a fitness coach, and if you get us a free gym membership, that would be amazing. And if you speak to us regularly, that would be great to just keep us motivated. So we developed a whole strategy. During the study, what we did is we had people kind of, we let them know what the study was about. They then registered or enrolled. They went and they gave us, they went to do a blood draw. They came in for a visit to our clinic there to make sure that they are allowed to and able to move their bodies without a risk of some sort of negative event, like a cardiovascular event or a cardiac event. We also had them complete what was called this Eco Week, where we had them rate their daily 6 times a day for 7 days before joining, before being randomized to the control group or exercise group. We asked them a lot of questions around their stress during the day and how they were feeling, how much control they had over their day. Once they completed all these parts, we also had them, we then randomized them to either receive 6 months of a free gym membership to a local YMCA. So if you lived in Berkeley, you got the access to the Berkeley YMCA, San Francisco, the San Francisco YMCA, and so on. Or they were randomized to a control group for 6 months, where we asked them, and they were all individuals who were already not working out. So we were like, hey, for the next 6 months, try to maintain your current behaviors, and in 6 months, we'll support you to become physically active. And then at the end of it, we then completed all the same assessments. This was with permission from one of our participants. This was our fitness coach, Andrew, from our team, who met the participant at the local YMCA, showed him his workout plan, and showed him how to use all the devices that we needed him to kind of wear while he was working out and so on. And what kind of plans did we give? So we started people off slowly, because that's the recommendation. People who launch in at 150 minutes per week often can't succeed in maintaining that, because it's really hard to maintain. So we start off at people like 3 times a week, 20 minutes per day per workout, at the gym or at home or for a walk, whatever it is. But we had them also have this like target of 40% of their heart rate reserve, which is at the low end of moderate physical activity. So a walk, pretty much. And then as they progressed through the study, we had them by the end of the 9th week, their goal was to work out 150 minutes per week, 4 to 5 times per week at the high end of their moderate physical activity. So that kind of goes between very brisk walk and a jog for some people. We also weekly, because they were wearing devices during their workouts, they weekly would upload their data. We would then send them this weekly report that said, congratulations on exceeding your 150 minutes. Well done, that's great. If they didn't complete the types of activities that we had set as their goal, the amount of minutes, we would also offer them, so even if they did 30 minutes, we say, hey, congratulations, you did 30, must have, seems like you maybe met some challenges, let's get on the phone, let's talk about this. So we supported them through these weekly phone calls with our fitness coaches who are trained in motivational interviewing. So I trained the students in motivational interviewing in order to support them to be physically active. We also sent them these 4 or 5 times per week text messages throughout the whole 6 months. So here's an example today is a great day for a walk. Meet a friend for a brisk walk instead of coffee or lunch. And we were trying to encourage them to get outside with people, develop some social connections through it, but also just try to have a positive frame on movement. So what were our results? So our first results that were published was on the primary outcomes of telomeres and telomerase. So telomerase is an enzyme that keeps telomeres long, and it was published in Psychoneuroendocrinology in 2018. First, just to highlight, our goal was for them to reach 150 minutes per week. And as you can see, as the weeks progressed, we had about a 70 to 80% success rate in keeping, in these participants, there were 68 of them in the 34 that were randomized, to be able to maintain 150 minutes per week, reach somewhere between 70 and 80%, depending on the week. If you set the limit a bit lower at 120 minutes, that was about 80 minutes or more, 80% or more weekly success rate, which is pretty significant since pretty much beforehand, people were at 0 minutes. What did we find? We had changes in their cardiorespiratory fitness. So exercisers increased in their cardiorespiratory fitness, whereas there was no change in controls. That's good, because if you move your body more, your cardiovascular system and respiratory system get stronger. We had changes in telomere length. So this is not the exact results, but it's the best way to put it in a figure. The exercise group increased in their telomere length over the 6-month period and was stable in the control group. Nothing changed for them. There were no changes in telomerase. And if anyone's really interested in the bio of like, how could telomeres stay long or get longer without changes in the enzyme that keeps them long? I'm happy to answer that in question period. And then we also had significant reductions in perceived stress and in the exercise group over 6 months, and compared to the control group who had no change. We next reported in another study some effects on our secondary analyses from people from all the participants who completed these questionnaires. We have no figures for these, but I'll just show you some highlights. Caregiver burden was the changes that happened in the exercise group. So this is what this treatment effect means. The changes that happened in the exercise group compared to the control group were different. That's what this confidence interval. So just ignore that, but the stars mean that it's significant. And so what happened in the exercise group was different from the control group. If we tease apart what happened in the control group versus the exercise group, you can see here that the control group decreased a little in their caregiver burden, but not significantly. There's no star here. But the aerobic exercise group, the ones who worked out, there was a significant decrease in their caregiver burden. There was also a significant decrease in their depressive symptoms, and also their sense that they're mastering life better. There was a sense of an increase in the exercisers that they increased in mastery. They had a better control over their lives. So that's the second paper that we published from this research. And then the third one was on daily life experiences, what I was telling you, that ecological momentary assessment week. So this is kind of how we completed the study. So at the beginning of the trial, before they were randomized to exercise, For 7 days straight, they received 6 daily questionnaires at different times throughout the day that was randomized. So we had a 42 questionnaires sent to them. It's high burden, but you get a lot of really rich data from these kind of studies. And during those times, we asked them, like, did anything stressful happen? Who are you with? Blah, 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 all these different questions. But we also had questions around like rumination. So have you been unable to stop thinking about stressful situations right now? When you were pinged, or at each ping that people got texted to answer these surveys, do you feel that you could control important things in your life today? They also rated how they were feeling: angry, anxious, embarrassed, sad, fatigued, and also positive emotions of happy, content, energetic, compassionate, and relaxed. And I'm just going to pull this up quickly and walk you through this. What you can see here, so these are the treatment effects on these rumination, control, and mood or affective states. These suggest that what happened in the exercise group is different than what happened in the control group over that 6-month period. And as you can see here, so this is the waitlist group, these first two in each category. This is rumination, this is controllability, negative affect, positive affect. In the control group, there was no change in their rumination, but in the exercise group, there was a significant decrease in how much they were ruminating daily. In the control group, there was no change in the waitlist group, there was no change in how much control they felt over their lives, but there was an increase on a daily basis when we pinged them and averaged across the whole 42 assessments. There was an increase in how much control they felt. There was an increase actually in negative affect in those in the control group and a decrease in negative affect. So that means it's better for the exercise group. So they had less anger, had less sadness, and so on. And some for positive affect, there was an increase in the waitlist group, but it was less serious or less extreme as the increase for the exercise group. So both groups actually went up a bit in positive affect, and we can talk about at some point, if you're interested, like how can you have negative affect go down and positive affect, or go up and positive affect go up, and that's something I'm happy to talk about for the control group if anyone has that question. But the exercise group really went up more significantly in their positive affect. So I'll just wrap up with, so there's a lot of other studies we've done in the lab, especially during the pandemic time with working out at home, using apps, looking at mental health primarily. If people are interested in that, I can send you papers that we've published for those. But the take home really is that exercise does improve these traditional and also novel markers of health in caregivers. It actually changes how we experience our everyday. And also what's really important is to understand our motivation, our barriers and facilitators for behavior change. It's kind of essential to start moving our bodies. We have to start understanding our own motivations, our own goals, our life goals, our health goals, what's stopping us from accomplishing, and how do we make our ideal self be more in line with our actual self. And on that, if anyone has questions, you can always email me at eli.puterman@ubc.ca. And thank you very much. Any questions from either on, I don't know, are you going to manage those? Okay. Are they typing them out, or are they? Okay, great. Any questions? Yes.
I think it's phenomenal that you found increases in telomere length with exercise.
Yeah.
I had one question. Was the baseline different in the exercise group compared to the control group?
No, so the way we did the analyses isn't really that way. It was using intention to treat, which is your typical way to do randomized trials. What they do is it predicts at the exact same time intercept and change over time. There were no differences in the intercept, but there were differences in the change over time. Yeah, so there were no differences then. Yeah. But so the increase in telomere length, there was no change in telomerase. And there could be a lot of other reasons. What keeps telomeres stable and long is the sheltering complex as well, which is a series of proteins that protect telomeres. So there is research showing that exercise can change the expression of the sheltering complex, all those proteins that are associated. Exercise also changes how many naive B cells we have circulating in our blood. And naive B cells, or which are younger, young B cells, naive B cells have the longest telomeres. So if you're kind of creating and exercise increases naive blood cells and takes out older cells from the blood, so you're washing out older cells and putting in more newer cells. So it could just be like the distribution of types of cells that we have in our blood. And we did look at the distribution of cells from your basic what's it called, the panel, CBC panels. So we did look at that, but we didn't have specific types of B cells or other types of cells. So yeah. Any other questions? Yeah.
When you sent out your the daily questionnaires throughout the day, did you adjust for the impact of sundowning?
Sorry, of which?
Sundowning.
Sorry, I didn't hear that word.
Sundowning.
Sundowning?
Mm-hmm.
I don't know that word, sundowning. Yeah,
I'll take you aside later. Then.
Okay, sundowning. Okay, I will look up that word. But I did not adjust for sundowning because I don't even know what that word is. So yeah.
When symptoms increase, typically at the end of the day, early evening, hence sundowning.
Oh, okay. Symptoms related to
Alzheimer's
disease. So we did not adjust for that. Thank you for explaining what that word is. I appreciate that. So this was on the caregivers, but of course that sundowning sounds like would probably have an impact on the caregivers as well. If you randomize people properly, you assume that whatever is occurring at baseline and 6 months later, in terms of the status of the family member, that there would be no real changes between the two groups, and over time, maybe as the disease progresses, there would be changes, but we would maybe think that those would be randomly also distributed. So the need to control for things in those kind of analyses when you have a randomized study is less severe, but it is something that I don't know if we even have how the severity of the patient. All we had was how many hours of care do you give? And so we know whether it increased over that 6-month period. Yeah, so I don't know. That's a great question. It's a great question. I'm going to look to see if we have any data on that. It's been a while that I've looked at the data.
We could talk after.
Yeah, I would love to. Thank you. Any other questions? Anyone online? Yeah.
Yeah, more of a follow-up question to the first question about the telomeres. If you can expand a little bit more on how we can lengthen telomeres, and then also, I guess, how exactly we can measure. Yeah,
that's a great question around telomeres. There's a very big push to try to understand how to lengthen telomeres. There's the few studies that have been done on exercise have shown that exercise can increase the length of telomeres. My study did not show changes in telomerase, but a study in a sample that had 5 times as many people as my study, I think they had over 300 people, did show changes in telomerase over a 6-month period. Also the mRNA, so the capacity or the amount of the protein or the mRNA associated with the protein production of telomerase has also shown to be increased in humans, I think over 300 humans, but then also there's so many studies in rodents showing that you can increase telomere length, sheltering complex that kind of protects telomeres. Reducing unhealthy behaviors. So there's research showing that different types of foods, especially the ones that we all think about, processed meats, can shorten telomeres. So kind of changing our diets has been shown. There's work on meditation and mindfulness showing that it can change telomere length. There was a book written about maybe 10 years ago, 8 years ago, called The Telomere Effect. It's a really great book written by Alyssa Epple and Elizabeth Blackburn on ways that we can change our telomere length. It's a book for the public, so it's also takes some liberties in extending sometimes some cross-sectional studies into suggestions of causal. So there are some criticisms of it, but it's a really phenomenal book in general, especially when they do highlight the interventions that are completed in this area of research. There are companies that sell you pills to increase your telomerase. It's all a bunch of fake companies from the States, but you can if you want. Yeah. Just press, yeah, now it's on. When you see red, yeah, now it's on.
Okay. I just had a question about like adverse experiences, just based on like your personal experience or knowledge.
I have a lot.
Yeah.
Sadly, yeah.
Do you know if children respond to stressors like differently than adults do? Like in terms of like being exposed to adverse experiences? Like how do they respond to stress? Like toddlers would often like tantrum, but like would that be like the same for like an adult? Like, I don't know.
Yeah, I mean, so I'm not a child psychologist, so
Okay.
there are different ways,
Mhm.
you know, but at the end of the day, there's some children who when they're exposed to stress, have almost like a meh, and some children have a more strong response. And there's research showing that there are some dispositional traits, genetic traits that kind of make some people more or less responsive to their environments. And those are called environmental susceptibility genes. So you can look up that stuff. But they really lead to whether people are more stress reactive versus less stress reactive. Those genes that make us sensitive to our environments have also been shown to, if within good environments, make you blossom and become a really great, you know, you're sensitive to your environment. So in a good environment, you're just thriving. So there's some work about like the dandelion versus the orchid. Totally off topic from the question that you had. It's my way of like, look over there, you know. In general, but like, I think a lot of it has to do with like how stress sensitive people are, and it might manifest in different ways. So some children have those tantrums, have the anger during these stressful moments. Some children kind of go into the corner and they seem to be adapting very well, but really they're having, you know, a horrible experience in the corner, feeling really, really sad, but they're not expressive in an anger way, in a fight or flight mode in that way. And then adults have their own ways of managing stressors. You know, we learn over time to use our frontal lobe and rationalize a little bit more and put things into context more, right? So we can maybe play it out a little bit better. But some people are still horribly affected by the stressors, but they kind of suppress their emotions in a unhealthy way that then leads to different health problems. So yeah, no problem. It's very complicated. Any other questions?
Just a few more from
Yeah.
online audience. Have you looked into any studies of how exercise improves focus and concentration, and if that's related to the decrease in cortisol level?
We are currently completing some studies. So we are currently completing some studies in the lab on how exercise at different intensities impacts stress responses in the lab. In studies not looking at how people are responding to stress right after exercise, in studies on attention and concentration, there's a really large, vast literature that people who right before completing a cognitive task, if they exercise, they can focus better and pay better attention to it. There's also a lot of research showing that people who exercise more, so not just in the lab you exercise people, but people who exercise more in general in their lives have better concentration and attention. What we're doing in the lab is a bit different, is that we're first getting them to exercise and then looking at how they're physiologically and psychologically responding to stress. We have shown in one previous study that different intensities have different cortisol trajectories, but this next study with a dampened approach, the more intense exercise, dampened cortisol response to the stressor. But we also have some the new studies adding onto that cardiovascular responses and psychological responses of appraisal. Like, will you assess a stressor more threatening or less threatening if you exercised beforehand versus not? Do you have less negative emotions to the stress if you exercised versus not? So we are kind of trying to unpack some of these questions around what happens immediately after you exercise in terms of stress responses.
And then one final question, I
Yeah.
think, just going back to the telomere stuff.
Yeah.
If anyone in general wants to know about their telomere length,
Yeah.
are there any available tests for the public?
Yeah, so there are available tests that you can spend a few $100s on. There are companies. So I don't know what those companies are. There used to be one in BC, and I know there's a lot in California and in the States. In general, though, there are no standards for like, this is the number that you need to have, and that is the most important thing. There's no cutoff the way we have cutoffs for diabetes, let's say, or we have pre-diabetes. There are no such things as or high diastolic blood pressure and so on. We don't have they don't have these kind of standards. And if a company tells you that there are standards, there aren't. But some people use this information to track their health regularly to see what is happening in their telomere length. Again, you can also just put on a cuff and look at your blood pressure. So I think those kind of companies are really trying to capitalize on people's excitement for this field of geroscience, that we can understand exactly what biological mechanisms are activated, and how can we manipulate those physiological mechanisms or biological mechanisms to our greatest capacity to increase our health span, not just lifespan, but increase our health span. But a lot of people are like capitalizing on this interest by creating these companies saying, we'll track all these novel biomarkers for you. At the end of the day, if we can stay healthy, reduce our stress levels, engage in healthy behaviors, those can probably mitigate a lot of the negative consequences, whether we have a biological outcome or our heart rate. Yeah. Well, it is 1:02, so I know some people have to run, and I have to run to go let someone into my building in an hour. So it was great to speak to everyone. Thank you to everyone who came online, and thanks to the group for inviting me, and thanks for everyone who showed up in person. Thanks.
Thank you, everyone, for joining. Thank you, Dr. Eli Puterman, for such an engaging presentation. I think if there's no more questions, we'll just wrap it up here. See you next year, and happy holidays, and stay warm.
Thank you.
Thank you.
Need to calendar.